In this presentation, we're going to look at the application of biological ideas, and we're going to focus on selective breeding of plants and selective breeding of animals. Animals include fish, of course, and we're going to look in particular at a syllabus statement which asks us to think about modern fish farming. In this slide, what I've done is I've grouped the syllabus statement so that you can appreciate that traditionally man has done selective breeding for many, many centuries to produce desirable characteristics within plants, desirable characteristics within animals. And what we do modern day is we take this to the next level. So here, this syllabus statement is going to look at the idea of micropropagation. And what we're doing in this case is we're taking a plant and we are cloning that plant. So we've got genetic copies of that plant in high numbers. And here, this is an extension again to look at how we can do this in terms of creating large numbers of commercially valuable crop plants. This is going to unpack the idea and story behind Dolly the sheep and cloning of an animal. Cloning of an animal includes taking a diploid nucleus and taking that diploid nucleus out of an adult and creating a brand new organism from that diploid nucleus. This syllabus statement is really cutting edge, and it looks at the idea of transgenic animals. And transgenic animals are animals that contain genes from two different species. And what we can do with transgenic animals is produce human proteins. This syllabus statement at the bottom is all about fish farming, and we'll unpack that in the next slides. So we're going to start off with the selective breeding of plants. And this is a great example. It's very similar to the one that's in your textbook, and it looks at how wild mustard, the original ancestral plant, has been deliberately manipulated by man to pull out desirable characteristics, and that allows us to produce all these different varieties of the brassicans. So the brassicans, all these cabbage, broccoli, cauliflower, Brussels sprouts, collards and kale, all come from a common ancestor, but they all look very different because man has deliberately selected them in a variety of different ways. Here, this is an ancient banana, and you can see that the ancient banana has got seeds all over the place, and we've selectively bred characteristics, seedless in this case, to produce this banana that we see on the shelves of the supermarket. So selective breeding is going to develop plants, and we want to develop plants to give us a higher yield, to have resistance to disease. So we're going to look at this idea of microbes causing disease, bacterial diseases and viral diseases in particular. Also, fungi can cause diseases in plants. And we've also got this idea that we want them to be resistant to pests. We also want plants to be hardier, and examples of hardier plants would be salt-tolerant plants. So if you've got flooding in many areas and you've got traces of salt in the soil, most plants can't cope with that. So we're going to select out plants which do have salt tolerance, and we can then grow them in these areas of the world. Frost tolerance is another example of a hardier plant. We also might want to selectively breed a plant which has got a higher nutritional value. Now, when we think about all of this, what we need to make sure that we do is we think about all the knowledge that we've met already about sexual reproduction in plants. So when plants reproduce, we need to take the pollen from one plant and we need to move it to the other plant. So we've got pollination. Now, what happens in selective breeding is we take the plant with a desirable characteristic and we take the pollen out of that particular plant and we move it into another plant that we know has got that same feature that we want to breed it with. So we've got fertilization in a very artificial sense in case of selective breeding of plants. If we look at this slide, again, this is showing you the same sort of idea as we met with the wild mustard. We've got the common ancestor here, which is some wolf-type species, and from that, man has selectively bred all these different varieties of dogs, all of which have got desirable characteristics. But in general, we want to selectively breed animals to create more meat, to create more milk. We want to have eggs that can lay, we want to have chickens rather that can lay more eggs, and to have high quality eggs, we want to have sheep which are going to develop fur, which is a very high quality. We can even link this to the race horses, and we want to selectively breed a horse which has got very, very fast running speed. Similar to plants, we can also look for the hardiness. So we can have animals which are growing in parts of the world which are not necessarily easy for animals to survive in. Again, I want us to connect what we already know about animal reproduction to this example here. So ordinarily, when animals reproduce, you've got the gamete of the male, which is the sperm, and that needs to be passed to the female. Now, in this day and age, what we do is we use artificial insemination techniques. So we deliberately take the sperm from the male and we inject that into the female to speed up this process. This video is on the Google Classroom, and I want you to have a look at that because that's going to underscore what all I've just been talking about, and it gives you lots more examples as well. This syllabus statement we need to unpack bit by bit, but what I want you to do is specifically go into page 211, pause this, read that, and come back to these slides. And we also need to pay attention to the fact that what we're learning here is connected to other parts of the course. So I want you to remember what we know about diet. And in order to grow, fish are going to need to have a source of protein. So in fish farming, we're going to make sure that the levels of protein are appropriate for that particular organism. We also want to think about this idea of ecology and trophic levels that we met previously. And in terms of controlling the quality and the frequency of feeding a fish, we want to link this idea to trophic levels and energy being passed along the food chain. I also want us to think about what we know from ecology in terms of intra-specific and inter-specific competition for resources. I want us to think about what we met with the blood system and the idea that our white blood cells will allow us to produce antibodies against antigens to control disease in our body. And we do this, we need to understand this in terms of the fish because we want the fish to be free of disease. We also need to think about the nitrogen cycle and in particular about decomposition because fish are going to produce waste, egested waste material can undergo decomposition and that can become very toxic if it's allowed to stay in the environment. We want to make sure that we can maintain the water quality within a fish farm by removing the waste products and preventing the buildup of things like ammonia within the water. We also want to connect the idea of selective breeding because we want to have a fish farm of fish which we know have got the qualities that we desire. It's probably worth pausing the video again here and just having a look at some of the labels which are on this diagram, but we can see there that there are a variety of different filters and that filter is maintaining the water quality. Also, what's important here to think about is the fact that some fish live in fresh water and some fish live in salt water. Now, in the example of salmon are very unusual because they can move from salt water and fresh water back and forth. But ordinarily, we've got fish which are bred either in fresh water or they're bred within the sea. This is a video again that I've posted in the Google Classroom, and I'd like you to watch that through because here he shows you some video footage, some actual fish farms, which is worth looking at. And as he says here, he's going to make a focus on the reduction of predation of the fish within these fish farms and the control of pests and the control of microbes and the prevention of disease. He's also going to talk about some specific examples of the diet fish are fed. This syllabus statement is looking at how we can use micropropagation. Micropropagation is sometimes called tissue culture. It's exactly the same thing. So we're going to take a plant that's valuable to us, and what we want to do is produce many numbers of exactly the same, genetically the same plants. Now you can read through these slides, these points rather, 1 to 7 for yourself, but I just want to underscore some of the things which are bolded up here. So we take the parent plant that we want to grow with desirable characteristics. And one of the things that we need to appreciate here is that we want a plant that is virus-free or disease-free to start off with. For number 2, what I want you to think about here is that the growing tip of the plant is meristematic. That means it can undergo mitosis. So the plant's growing tips, the root and the shoot, in fact, are meristematic tissue. It's tissue that can undergo cell division by mitosis repeatedly. So that's the part that we need to remove. And just to make you appreciate that meristematic tissue in plants is exactly the same as a stem cell, which is within an animal. So they're undifferentiated and they retain their ability to divide. So we're going to take the plant, we're going to remove the growing tip, and we're going to make sure that that growing tip is sterilized because we want it to be free of any particular disease. And then we take it and we basically mash it up. We mash it up, we add it into a Petri dish, and the Petri dish contains an agar jelly substance. The agar jelly substance is going to provide a growth medium for those plants to do cell division. We're also artificially going to have plant hormones within that growth media, and the plant hormones are going to stimulate the plants to undergo cell division. And what we get is a big mass of cells, all of which are genetically identical. We're going to remove some of those cells. Again, we use very, very specialized plant hormones, also called auxins. And this time, the auxin that we're going to use is going to cause the plant to undergo differentiation. So that under differentiation is going to grow into small explants, and those small explants can be grown, oftentimes they're grown into the glasshouse. This is a diagrammatic version of everything that I've just said, and this is the video that I've posted in the Google Classroom, which again I want you to watch because he unpacks this step by step. This is the next thing that I want us to consider, this idea of why do we want to do micropropagation. There are advantages and there are some disadvantages. The advantages are kind of self-explanatory. Really, we can produce large numbers in a very short space of time. That means that we can produce more crop and we can have more commercial gain. They've all got the traits that we want. They're all disease-free. What we can also do is we can undertake this example of micropropagation with endangered species and make sure that we keep that species and grow it in conditions where it can thrive. Also is this idea that we don't need to have pollinators, it's all artificial, so we can speed it up and we can do it at different times of the year. So we can grow plants in the winter where ordinarily they would only grow in the summer. We can also do this with genetically modified plants, which we'll come back to later on. The disadvantage is that they are clones. They're all genetically identical, and if they're all genetically identical and a new disease arises, then all of those plants are going to perish from that same disease. They're all going to also be non-resistant to the same pest. So just bear in mind that disease here, we're talking about microorganism. Pest could be an insect pest, could be a fungi pest, so they're not necessarily the same thing. Also, a disadvantage is that the lab has to be super sterile, because if it's not sterile, then the crop will fail, and it's an expensive technique, so it's very costly. So this brings us to halfway through this slide. I'm going to pause there, and we'll carry on next lesson looking at the story of Dolly the sheep and cloning of animals. But this is all about how we clone a plant.